What if I told you we can actually rewrite the blueprint of life? Well, there's a powerful technology out there that lets scientists do pretty much that, to turn genes on and off, to figure out all their secrets. So let's dive into the revolutionary science of gene knockout.
You know, for as long as people have been growing flowers, the blue rose has been the stuff of legend. It's always been this symbol of mystery, of the impossible. Why? Because in nature, it just doesn't exist. And yet, here we are, looking at one.
So, that's the big question, right? How did we get from total myth to actual reality? If nature said, nope, can't be done, how did science find a way to make it happen?
Well, the answer is hidden deep inside the rose's DNA. Scientists basically hacked the flower's genetic code. They went in and turned off the genes that were making red and orange pigments, and then they added a whole new gene to create blue. That process of turning a gene off, or knocking it out, that is the key to this whole story.
And you know, this whole idea of a genetic off switch, it's not just for making beautiful flowers. It's actually a fundamental tool for understanding the book of life itself. I want you to think about it like this. Your DNA, your genome, it's like a gigantic, incredibly complex instruction manual. But here's the catch. You have no idea what most of the instructions, the genes, are actually for. So how on earth would you figure it out? Well, you'd probably start by, I don't know, deleting one sentence at a time and then just seeing what changes, what breaks. And in biology, that process has a name, gene knockout. It's a technique where scientists intentionally disable a specific gene just to see what its purpose is.
The logic is actually super simple and powerful. If you pull a spark plug out of a car engine and it suddenly won't start, well, you've just learned what that spark plug does. Gene knockout applies that exact same thinking to the machinery of a living cell.
And make no mistake, this is not some minor lab trick. This is a huge deal. The scientists who pioneered this, Kopecky, Evans, and Smithies, they won the Nobel Prize for it back in 2007. This discovery didn't just push biology forward, it fundamentally changed how we study life itself. It gave us a real way to prove cause and effect for a single gene.
Okay, so that's the theory. But how does this actually work in a lab? Let's walk through the classic example, the making of what's called a knockout mouse.
All right, it all starts with embryonic stem cells. These are incredible. They're basically the body's master cells, these blank slates from an early embryo that can become any part of the body. So scientists start by taking some of these from a black mouse.
Next up is the actual knockout. Using some really sophisticated molecular tools, scientists introduce a custom-made, broken version of the gene they want to study. The cell has this natural repair process, and sometimes it'll swap its own working gene for the new, non-functional one.
Now, this is where it gets really clever. Those modified stem cells are carefully injected into a totally different embryo, this one from a brown mouse. The different fur colors aren't just for looks, by the way. They're a built-in visual tracker so the scientists can see what's going on.
So this new mixed embryo gets implanted into a surrogate mother, and if everything goes to plan, a chimeric pup is born. This little pup is a living mosaic. Some of its cells are normal from the brown mouse, but others from those black mouse stem cells are now carrying the knocked-out gene. And that patchy coat is the very first sign that the whole experiment is working. Now that basic technique was absolutely revolutionary, but it wasn't perfect. It had some serious limits. And of course, science never stands still. The next step was to build a smarter, much more precise genetic off-switch. So here's the problem. What if the gene you want to study is essential for an embryo to even develop in the first place? If you knock it out from the very beginning, the pregnancy could just fail. That made it incredibly hard to study genes involved in basic survival or diseases that show up later in life. To get around this, scientists came up with the conditional knockout. It's a much more sophisticated approach that lets them turn off a gene not everywhere, but only in a specific place, like just in the brain, or at a specific time, like only in adulthood. One of the most elegant ways they pull this off is with something called the Kree-lock system. Think of it like a two-part security system for DNA. Scientists put these special DNA tags called LOXP sites on either side of the gene they're targeting. Nothing happens until they introduce an enzyme called Cree. Cree acts like the key, activating a pair of molecular scissors that only cuts the DNA right at those LOXP sites, popping the gene right out.
Now I know this all might sound like super complex science that's trapped in a lab, and it is, But the discoveries that have come from these techniques have a direct and honestly a profound impact on our understanding of human health. But first, a quick clarification, because you might hear another term out there called knockdown. A knockout is like completely removing a light bulb. It's gone. A knockdown is more like using a dimmer switch. The gene is still there, its activity is just turned way, way down. Both are useful, but they're different tools for different jobs.
So, what have all these knockout mice really taught us? Well, just about everything we know about the genetic basis of disease. By turning off one gene at a time, we've created mouse models that mimic human conditions like cancer and Alzheimer's, letting us study them in ways we never could before. For instance, knocking out the p53 gene in mice helped us confirm its vital role as a tumor suppressor. We've used these models to understand the genetics of aging and memory, and they're absolutely essential for testing new drugs safely before they ever get to human trials.
And why mice, of all things? Well, here's the number that will blow you away. About 95% of the mouse genome has a direct counterpart in our own human genome. That incredible similarity makes them an almost perfect stand-in to study human biology and disease.
And all this technology brings us full circle, right back to that idea of a chimera, an organism built from more than one set of DNA. What started out as a clever lab technique has now blown open these new frontiers that start to blur the lines between species, pushing us into territory that is as fascinating as it is ethically complicated.
Which leaves us with a final and pretty provocative thought. This ability to turn genes on and off gives us a level of control over biology that is completely unprecedented. We've gone from just observing life to actively rewriting it. So the question isn't, can we anymore? The question is, should we? And as our power grows, where do we decide to draw the line?